so for ecology, back to our lecture materials here, we ended up on Monday by talking about net primary productivity and terrestrial biomes, right? So I told you guys you don't have to know all of the characteristics of each of these biomes on the list, but I do want you to be able to talk about sort of, is it a wet habitat or a dry habitat? Is it temperate or is it hot or is it cold? So you don't really have to know the things like you see in some of these descriptions about, you know, degrees of latitude where you find it, or necessarily even where in the world specifically. Like, I'm not going to ask you to put the chaparral on a map or anything crazy like that, but just sort of be able to talk about or answer questions about the relationship between latitude and net primary productivity, essentially. So location of biomes, what that means for productivity, that's basically it. General characteristics of these terrestrial biomes. You guys all good with that? Makes sense? Okay. So what I want to do today is finish up this chapter. It won't take very long. We're going to talk about aquatic biomes. So we will talk a little bit more about the characteristics of our aquatic biomes, more so than we will with the terrestrial ones, just because they're a little bit different and interesting. So we'll go through marine biomes, estuaries, which is kind of the middle ground between marine and freshwater, and then we'll look at some freshwater ecosystems as well as we move through the rest of this information here. And this is a nice little map, again, kind of like the terrestrial biomes map, just showing you where you find these different types of habitats globally. And of course, this is not comprehensive because you're looking at the entire world, right? So it's major habitats. There's of course going to be micro habitats all over the place. Speaking of, you know, when we're talking about water and aquatic biomes. So the first one we'll look at is the ocean. So the ocean is definitely marine, right? When you see the term marine, what does that mean? You guys know? Marine versus freshwater would be the comparison that you usually see. Yeah, salt, right? So we're definitely talking about salt water. We're talking about the ocean. What you guys will be expected to do with this is to be able to recognize mostly, here's the key in the red box here, in which of these zones would you expect to see photosynthesis, and sort of what would you see elsewhere? Okay, so when we're looking at the way you divide up the ocean, you've got the pelagic zone and the benthic zone. Pelagic means open waters. So when you talk about something like pelagic seabirds, you're talking about things like petrels that live out on the open sea. Okay, so pelagic is out in the oceanic zone, okay, way out over the open waters. And then benthic is the bottom. So you can have benthic in any aquatic biome. Talk about the benthic zone of a lake. It's fine. You can use that terminology. But just know that benthic means the bottom and pelagic means open waters. Okay, so just a little bit of ocean terms. As far as the zones of sort of distance from shore, the intertidal is one of the most interesting zones, in my opinion, for looking at adaptation. Because the intertidal is basically the beach. Okay, why do you think it's called intertidal? What's the tide? What happens to the tide? That comes in, goes out, right? How far up the beach the water comes changes at least twice a day, right? Based on cycles of the moon and gravity and all sorts of different things. So if you live there, you've got to be pretty tough, right? You've got to have a pretty good set of adaptations that allow you to both sustain drying out, right, or getting wet, and also constant pounding of waves, right? So if you live in the intertidal, you have to be able to sustain that mechanical disturbance all the time. So thinking about the zones, it's interesting, kind of, but when you start to think about who lives there, it becomes more interesting from an ecological standpoint, right, and adaptation. So that's intertidal. The neritic zone is from the intertidal to the edge of the continental shelf. So usually the continental shelf, what happens is you have sort of shallow waters out to about a depth of 200 meters, and then it drops off, okay? And the shelf can be miles away from shore, or the shelf can be right offshore. So different places are going to be different in terms of how far out the continental shelf drops off. But this zone from like beach to about a depth of 200 meters, that's what we call the neritic zone. Would you expect to see photosynthetic organisms in the neritic zone, do you think? Based on, you can look at the sort of picture here and see what you can conclude. Is it shallow? Sorry? Is it shallow enough to have light penetrance, do you think? Yes. Yeah. So would you expect to see photosynthetic organisms there? Yeah, for sure. You'll notice that we're also calling about 200 meters the photic zone. Okay, if you guys are working on water, you know what the photic zone is. If you're not, you can probably guess. What does photic mean? Light, yeah. So light can penetrate, right? So what do you think aphotic zone means? No light or very little, right? It's getting down there. So you're going to see absence or decreased photosynthetic organisms in the aphotic zone. And then you get to the abyssal zone, which is depths below 4,000 meters into the abyss, right? It's dark, it's cold, no light penetrates. And this is the zone where you often will see those hydrothermal vent communities where all that really weird stuff lives, right? Like yeti crabs and chemoautotropic bacteria and stuff like that. Right? So really different types of communities exist in these different zones. But what I really want you to sort of think about is that, right? So distance from shore, adaptations for water movement, light penetrance. So I do want you to be able to recognize these definitions, but I also want you to think of why we're talking about it, right? Not just so you have to memorize these things, but also because it is, we're talking about, ecology. So we're just basically introducing these ecosystems so you can start to put things in context, okay? All right, coral reefs deserve their own discussion because they're one of the most diverse habitats on the planet and also one of the most sensitive, right? So coral reefs are basically the remnants of past cnidarians. So remember that corals fall into the same group as jellies and hydras and things like that. So they do have the ability to use cnidocytes. You guys remember what cnidocytes are? What is cnidarians named for? What did jellyfish do? Yeah, they're sting, right? They sting to catch prey. So cnidocytes are those stinging cells. Coral are not necessarily always going to do that. They do have that relationship, that symbiotic relationship with zooxanthellae that we talked about back in the Protists chapter, kind of like forever ago. But remember, those are those single-celled organisms that live inside the tissues of the coral polyps and give them food, essentially. So the zooxanthellae are photosynthetic, and they're giving the coral polyps sugar. And so if those species of coral that have that symbiotic relationship tend to live more shallow in the more shallow waters, right? Why does that make sense? If you have a photosynthetic symbiont that's feeding you, why would you want to live shallow? Sunlight, yeah, light penetrance, right? So as you get a little bit deeper, you often see those coral that do have the cnidocytes that actually sting things to capture their prey. So you see both ways of doing things, both types of sort of life strategies in these coral reefs. When the coral are living, they make calcium carbonate exoskeletons. Okay, so they secrete their own sort of hard exoskeleton. And they use calcium from the water and carbon monoxide to build it, just like any other type of sort of shelled organism. But when they die, they leave those behind and they pile up on each other and they build these reefs, right? And so the reef itself becomes habitat for hundreds of other species. Fish and anemones and all kinds of stuff use the reefs for cover, which makes coral what we call a foundation species. Have you guys heard the term foundation species before? I don't think we've talked about it yet in here. A foundation species is a species that provides the actual physical foundational habitat for everybody else that lives in a particular community. Okay, so sometimes you may talk about like our hypothetical forest that we were talking about with the white pines and the hemlocks and everything last time. You could consider the trees a foundation species in a forest. You could consider kelp a foundation species if you're talking about a kelp forest in the ocean. Coral is a foundation species because it builds the reef, right? So if you take the coral out of the equation and you have degradation of the reef system, you lose habitat for all those other species. And because of where you find coral reefs, warm, shallow, tropical type areas, biodiversity tends to be high. Excuse me, why is biodiversity high where it's warm and sunny? What did we talk about last time? What's that major concept that we spent so much time talking about? What happens where it's warm and wet and nutrient levels are high? Net what? Productivity. Yeah, primary productivity. So biodiversity is going to be correlated with that. So you see a ton of species using these reefs, but the problem is that reefs are super sensitive. Remember, I don't know if you remember, we talked too much about it when we were talking about that symbiotic relationship between zooxanthellae and the coral polyps, but when the coral gets stressed, they tend to kick those zooxanthellae out. You guys remember that? We talked briefly maybe about coral bleaching. So we call it coral bleaching because they often lose their color, because the coral polyps themselves are kind of clear and colorless, but the zooxanthellae that live inside are photosynthetic. So they've got photosynthetic pigments, which give them an array of different colors. So when that coral polyp gets stressed, which it does really easily with changes in environmental conditions, it kicks them out. And if they leave, the coral isn't getting food anymore because it's dependent on that coral, on that symbiotic relationship with the zooxanthellae for food. So it doesn't really make sense. And we don't really understand yet why they do that. We just know that they do. So when you see a bleached coral reef, that is an indication that the ecosystem is in danger, right? That things are going on that are causing problems for those coral. So what kind of changes? Well, they're sensitive to changes in pH. So ocean acidification, right? Which is a big deal with climate change. Warming temperatures. So they live in this sort of nice, warm range, but they sort of exist at the upper limit of comfort for temperature in organisms. Okay, so it's nice and warm, it's really warm. If it's a little bit warmer than that, it's too warm. Does that make sense? So they're like upper limit of temperature as it is kind of their sweet spot. But if you get any hotter, then they're super sensitive and they start kicking out their symbionts. Okay, so coral are really sensitive to the types of changes that go on with warming climate and ocean acidification, but also just pollution. Their chemicals make them irritable, right? They're just very, very sensitive. So not only is it important because they're a foundation species and they're providing habitat for all these other organisms, but they're also an indicator, right? A bioindicator, a species that lets us know that there are problems in that particular ecosystem. Okay, so that's why coral are important, coral reefs are important habitats to talk about as far as aquatic biomes. Good? Moving on. Do you guys know what an estuary is? I said it was kind of a middle ground between salt and fresh, right? An estuary or an estuarine habitat, you may hear that used as an adjective, is where a source of fresh water meets the ocean. Okay, so a river dumps into a delta, that's an estuary. So you've got what we call brackish water, and brackish just means dilute salt water. So it's partially fresh, partially salt, it's mixing right there, right? So you can imagine a fresh river flowing into the ocean, right where they meet, you're going to just basically have that mixing. You're going to have diluted salt water that's brackish. It is really good habitat for lots of organisms. Nesting habitat for lots of crustaceans. What are crustaceans? You guys remember? Crabs and yeah, there are arthropods with exoskeletons, crabs, shellfish, stuff like that, right? What are those little crawdads? Those little crustaceans, mollusks, right, like snails and slugs, nesting habitat for fish, great for shorebirds and wading birds. So birds that eat things like crustaceans and mollusks will also nest around this area. So you get a lot of biodiversity in estuarine habitats. But plants and animals that live there have to be adapted to salinity. What is salinity? Do you guys know what saline means? Salt, I mean, it's salty. Yeah, so salinity just means degree of saltiness, how much solute is dissolved in that water, and salinity changes with the tides. So again, an estuary habitat is really similar to an intertidal type habitat because you have those tides that come in and they come out. So I want you guys to think about what's going on. If you are at the mouth of a river that's meeting the ocean and the tide comes in, what is happening to the salinity level right there at that estuary? It's increasing, right? Why? Yeah, because the tide coming in and this tide is ocean and the ocean is salty, right? So the salt salinity is going to increase when the tide comes in. So what happens when the tide goes out? What do you think? Salinity is going to decrease because remember you've got flowing fresh water now in the opposite direction, and so it's going to be constantly fluctuating. Well, how about you and me? How well adapted are we to changes in salinity? What do you think? We have systems in place in our bodies to make sure that we stay salt water balanced? Yeah, if you get too salty, you become dehydrated, right? If you have too much water, that can be a problem too. So we have a super narrow range of salinity that we can like operate in homeostasis. But these organisms that live there have to be able to tolerate those fluctuations. So again, you're looking at really interesting adaptations that allow them to do that. Plants have to be halophytes. Halophytes just mean plants that like salt. Remember halophiles from bacteria? It's been a while, right? But halophilic bacteria or halophilic archaea, prokaryotes that can withstand extremes of saltiness. Okay, so halo means salt. So you've got marine, that means salt water. You've got salinity, which means level of saltiness. And you've got halo, which is another prefix or root word that means salt. Okay, so all these have to do with marine habitats. Halophytes are just plants that can tolerate salty conditions. They have adaptations like things like root filters that remove salt and things of that nature. So they're well adapted to where they live. Animals that live here also have to have adaptations to the changing of the tide. So a really cool example is those mussels and clams, those bivalves that can actually close their shell. What they do is they stop feeding and close up the shell tightly. If you've ever tried to pry open a shell, a bivalve, they have incredibly strong muscles that are holding those two sides of that shell closed. Right? If you ever, anybody, I don't eat them, but anybody eat them? Like oysters or clams? But anyway, I've seen people do it, right? Where you like take that tool and sort of pry it open really hard. You guys know what I'm talking about? Yeah, it's because they have these incredibly strong muscles that contract to slam that shell shut. Well, for one thing, it's protection, right? Because other organisms, not just people, want to eat them. Okay, so they can close their shell to protect themselves from predation, but these guys who live here can also do that to keep water out. So when the tide is low, they will slam shut their shell and switch to anaerobic respiration. So let's think about how this works. When the tide is low, what is happening to the water, to the salinity? It's going down, right? So more fresh water is rushing in. So let's just pretend for a second that you're a mussel and you're well adapted to salinity. What happens if you leave your soft tissues wide open and fresh water rushes in? What do you think? Think about osmosis and diffusion. Everyone loves to talk about that, right? If your cells are salty and you're well adapted to being sort of isotonic with salty water, what happens when you're doused with fresh water? Which way does water move? Into the cell or out of the cell? It's going to move in, right? Because water follows solute. So if you're salty and you're well adapted to salty, and then all of a sudden it gets fresh, water rushes into your cells, what happens? Yeah, lysis, explode, right? They're gonna split. So these guys just slam shut and they switch to anaerobic respiration. So these guys are aerobes, they use oxygen for cellular respiration, but they can switch it off. Because if you're slamming shut to keep water out, you're also keeping oxygen out, right? Because where are they getting their oxygen? From the water, right? So they can actually switch their respiratory or their respiration style, so to speak. It's pretty cool to think about. So these are, although it doesn't seem like the most exciting topic, talking about estuaries, there's actually some really cool biology that goes on in that zone because you have to be well adapted to those changes. So there you go, estuaries. Lots of biodiversity, lots of cool stuff to see. All right, freshwater habitats. You guys know these already. This is why everyone likes ecology. It's stuff you've heard of before. Lakes and ponds versus rivers and streams. What is the difference between lakes and rivers? One moves, the other doesn't. Yes, largely. Okay, so lakes and ponds are standing water. They can be small, as we know. They can be huge as well, right? So Lake Superior is the largest freshwater lake in the world. Where's Lake Superior? Do you guys know? Generally. Like, North, yeah, North America, right? It's here, where it's on our continent, which is a kind of cool thing to be able to say. 82,000 square kilometers. Lake Allatoona, which some of you are pretty familiar with, 48. So Lake Allatoona isn't tiny. It's not like Delaware Lake, right, out on the preserve, which is like teeny. Yeah, so Allatoona is not like the smallest lake you've ever seen, but compared to Lake Superior, that makes the Superior look like the ocean, right? So they vary widely in size. And lakes can have photic and aphotic zones as well, right? So remember the photic zone is just where light can penetrate. So the deeper your lake, the more sort of aphotic zones you may have, but they both exist. Rivers and streams, again, moving water coming from a headwater, which is the source of the water. Usually it's going to be, collecting rainwater that's sort of trickling down in tiny little rivets until you hit little tiny streams and creeks that then join up into larger bodies of water that get bigger and bigger as they flow towards their mouth. So you got Nile, Amazon, and Mississippi are the largest in the world. So we actually brag about one of those as well here on our own home continent. There's a channel, that's where the water is moving, right? That's sort of the middle. It's narrowest at the source and widens as it travels. So usually water slows down as the channel widens, right? Narrows, it starts to move a little bit faster. And there's usually less sediment near the source. What's sediment? You know what sediment is? Like stuff in the water. Things that get deposited, dirt, runoff, stuff like that. Particles, particulate matter. Less near the source as it's first picking up, and then more as the current gets slower. So it tends to be more dusty and silty where the current slows down. So this isn't anything groundbreaking. You guys know the difference between those, I'm sure. Okay. So that gets us to wetlands, which is our last habitat that we'll talk about really quickly here. What do you think wetlands are? Why do you think they're called wetlands? Because they're usually covered in water. They're wet. See, ecology, right? Terminology is so much more straightforward in this unit. Soil is permanently or periodically saturated with water, which again, you're going to see adaptations to that sort of habitat. So you have to have vegetation that can withstand their roots being constantly inundated with water. Oftentimes the roots themselves are completely underwater and the soil is totally saturated, right? So there's water all the way up and above the bottom of the trees and shrubs, but portions of the shoot system come up out of the water. That's called emergent vegetation. This means it's emerging from under the water. Things like marshes, bogs, swamps, mudflats, salt marshes, these all qualify as wetlands. So it's just wet. And it's wet most of the time. Marshes and swamps are going to form through slow and steady water flow. Okay, so very, very slow, gradual flow of water. Even something like the Okefenokee Swamp. You guys ever been to the Okefenokee or you know where it is? Where's the Okefenokee? Right there. You guys gotta go. It's in Georgia. It's our big swamp. It's like huge. Like the lower half of the state is the Okefenokee Swamp. Really huge swamplands, but formed because it's a basin. Like it's a low spot in the land where you have just water that very slowly runs into it. So even though it's a swamp and it doesn't flow like a river, there is actually current in some places in the Okefenokee Swamp. Also, lots of really cool stuff lives there, you guys. If you ever get the chance to check it out. Another type of wetland is a bog, and bogs are going to form where water flows hardly at all. So sometimes, most frequently, you get these from what we call seeps, where there's like water coming up from the ground, from spring water underneath. It doesn't have anywhere to go because there's a depression also in the earth. So water collects there, but it's stagnant. What does stagnant mean? Do you guys know that word? Still, yeah, it's not moving. It's not, there's no flow to it at all. Okay. What happens in bogs is they tend to be nutrient depleted. That means there's not a lot of nutrient availability in bogs because stagnant water is usually low in oxygen because you need movement of water to bring oxygen in, right, to mix it into the water. Decomposition of organic matter is slow because there's low oxygen, causing buildup of the organic acids that are a byproduct of decomposition. Lower pH, right? Acid is going to lower the pH of the water, is going to lower nitrogen availability because mostly nitrogen fixing bacteria don't operate really well at low pHs. And so you basically end up with low net primary productivity. It's not that healthy, not that nutrient rich of a habitat, but you still see things that live there, things with really cool adaptations like carnivorous plants. You guys know what carnivorous plants do? Yeah, they eat insects. So if you live in a bog where nutrients are limiting, now this is an example. This is a pitcher plant, Sarracenia. genus. Also things like Venus flytraps. You guys know what Venus flytraps are, right? Like the leaves that catch stuff like this. Yeah, they're still photosynthetic. You guys with me? So they're making what? You're photosynthetic, what are you making? Making your own food. What kind of food, right? Sugar. Yeah, sugar is organic. Yes? What about something like nitrogen? Nitrogen is not something that a plant can make, right? It can make sugar all day long, but nitrogen is often a limiting nutrient for plants. They get it from the soil. That's why they depend on nitrogen-fixing bacteria, right? To make that nitrogen available. Well, if that's not happening in the bog habitat where you find these types of plants, they are supplementing their nitrogen by eating insects. What is it chemically, you may or may not know this, but I'm going to see if you do. What is it chemically about the insects? What do insects have in common with something like a cheeseburger as far as nitrogen? What's it made of? What does meat primarily consist of? Protein, right? And what are the building blocks of protein? Amino acids. And what makes an amino acid an amino acid? Nitrogen. Yeah, the amine group, right? NH2 on the end. You guys remember that? You're like, God, I hate it when she makes me think about 11 and 7. Yeah. So they're high in nitrogen. Well, to these guys, a fly is basically like a steak, right? Source of nitrogen. It's an animal and it's built of protein. Yes? All right. So that's an adaptation to life in a nutrient depleted bog. So it's cool if you think about why different things live where they live. Right? It's kind of boring if you just look at the definitions of the biome, but when you start to think about the big picture and sort of what are we talking about here? We're talking about ecology, organisms adapted to their environment, and then it gets a little bit more interesting. Okay? All right. That ends the intro chapter on ecology. So that's your foundation of what we're talking about, big picture issues that we're talking about going forward. You guys have questions from this chapter? Anything you want to talk about before we move on? Everybody's good? All right. Moving on. Next week, we will do a Kahoot called Name That Ecology. You'll get to tell me, I think I mentioned this, that you guys will have problems or questions on your exam where you'll have to tell me if it's population ecology or community ecology or ecosystem. We'll do a little Kahoot next week to sort of go through some of that stuff. But that's what we're going to look at in chapter 45. Chapter 45 has a ton of information in it, so it's a little bit longer PowerPoint. It'll take us a couple days to get all the way through this chapter because population ecology and community ecology are two related but different topics, right? So there's a lot of information to talk about here. Basically, what we're going to look at in this chapter are ways that you can ask questions in each of these branches of ecology. Okay, so we're going to look, and remember I told you we'd look at a bunch of graphs, measure things, count things. We'll look at how ecologists study what they study. So we'll get to talking about Asian carp towards the end of the chapter, but there's also in the D2L module for this chapter, there's a link to a video about Asian carp, and it's kind of interesting. So if you have the opportunity to look at it ahead of time, then you'll have a little bit more foundation and background when we get to talking about Asian carp. But these guys are an invasive group of species of fish. Okay, so in this chapter, we'll discuss invasive species and what that means, because we'll get to community ecology, which is interactions between species, right? So what does it mean when a species is introduced outside of its native range and it can outcompete the native species that live there? Okay, so we'll be talking about that a little bit later. That's why that's used to introduce the chapter. But before we get to that, we're going to go all the way through population ecology and look at some of the ways that we measure and ask questions in this particular area. Okay, so remind me, what is a population? We talked about population, we talked about organism, population, community, and ecosystem. What's a population? Oh, it's not even on there. A group of organisms. They can interbreed because they're the same species. Yep. So a group of organisms, all the same species, interacting, interbreeding, that's a population. So population ecology studies how populations interact with their environments. When we start talking about how different populations interact with each other, then we're talking about what? Community ecology, exactly. So yeah, they're intertwined. And remember, when you guys have to make your case that a particular study is population ecology or community ecology or whatever, there may be more than one right answer. And all you have to do is say, I call it population ecology because, and then give me some evidence. Okay, so there's always going to be interconnectedness between population and community and even ecosystem ecology. Populations are dynamic. Do you guys know what dynamic means? Does it mean that they stay the same all the time? No, it means they're always changing, right? So populations are made up of living organisms, and living organisms are always changing, and the environment is always changing. So populations are always changing. Some things that we look at in population ecology are population size and population density. These are different but related quantifications that we can do. Population size is easy. It is just how many individuals are in that population. How big is it? How do you measure population size, do you think? Like basically, you count. Yeah, exactly. There are going to be instances where you can't count the entire population, right? Because it's too big, or it's widespread, or you can't catch all the squirrels, for example. So you can sort of make estimates and take samples and do extrapolations and things like that, but you're counting essentially. So population density is related in that it is still a number of individuals, but then you add in this piece, per unit area. So what are we looking at here? You're looking at the number of individuals, but you're also looking at how much space they take up. The density refers to how many are packed in or how many are widespread. Okay, sparseness versus, I guess, density would be the opposite of sparseness. The number of individuals per unit area, how many live in a square meter, how many live in a square mile, how many per hectare, doesn't matter. You're just talking about a unit area. So if you're looking at a population of bacteria, you could potentially talk about per milliliter, right? So your unit area can be really, really tiny. Or if you're talking about something like, I don't know, population of elephants, your unit area is probably going to be bigger than the milliliter. Yes? You guys with me? So you, once again, as a population ecologist, you're setting your own parameters based on what you're trying to study. Okay? Distribution. We'll talk a little bit about some species distribution patterns, which is looking at how individuals are spread throughout their range. And you guys remember what the range is, right? We talked about this last time. Just where in the world, geographically, a species is found. Okay, so we'll look at range maps. We'll talk a little bit more about that. But within the range, how do the organisms use that space? Do they clump together? Do they defend individual territories? What are they doing? That's species distribution patterns. So we'll look at those. And then we'll look at demographics. This is the statistical study of populations. This is where you count and look at things like how many are born in a year, how many die in a year, what's their average lifespan, what is the age when they first reproduce, how many young do they have at a time. All of those things are going to be measurable statistically. Okay, so we're going to take a look at that. Tons of fun. Aren't you excited? I know. Everybody loves this part. All right, so let's look at some measures of size and density and why it matters. So first of all, genetic variation. How do you think population size can affect genetic variation in general? Yeah, a too small population can lead to a genetic bottleneck. Like, what does that mean? What happens to genetic variation if you have a bottleneck? Decreases, right? So you can look at things like inbreeding depression and stuff like that, right? We talked about that many chapters back. Essentially, more individuals, usually, not in every case, but in many cases, means more alleles. So more alleles means more genetic diversity. It means more variation in phenotype, which means more adaptability, which ultimately, usually, means a healthier population, right? So that's going to be one of the basic reasons that you look at population size. High density, however, can be bad, right? If you have a huge population in a small area, what is happening to competition? Huge population, small area. Competition is going to go up, right? Because you're competing for space, resources, mates, who knows, everything, right? And then low density can also be detrimental if you are a cooperative species and you need others. What if you can't find a mate because your population is so widespread and sparse? So these are the kinds of things that we think about. There is an inverse relationship between body mass and population density. Now, this is our first example of the types of graphs that ecologists love to make. And this looks really complicated when you first take a glance at it, right? Kind of. But it's actually super easy, you guys. What does an inverse relationship mean? When one thing goes up, what happens to the other? Down, and vice versa, right? So the inverse relationship between body mass and population density means what? If you are bigger, you have more body mass, how many individuals can fit in a particular unit area? Fewer or more? Fewer, right? Yeah. That's all that means, you guys. That means if you're small, you can squeeze more into the same space. That's what that means. So this nice little graph here, we're looking at density of population per square kilometer and mass in grams. But this is a log scale, which makes it a straight line. Okay? So you don't have to worry about that too much. But what's basically happening here is you've got this nice list of Australian mammal species. And the smaller you are, let's see, here's your mass in grams, start here at the low end. The smaller ones tend to have more density per kilometer square. And the larger ones, things like wallabies, gliders, bigger, fewer in the same area, kilometer square. Okay, so that's pretty straightforward. This is the kind of stuff we're looking at, you guys, okay? With this. All right, so that's why we look at size and density for a couple of reasons. Distribution and range. So this is the same thing we introduced in the first slide about what are species distribution patterns, but here are some examples. Okay, I'm definitely going to give you scenarios and you'll have to tell me which one is which. Okay, so let me also continue to remind you, just like the case study questions that you'll see about what type of ecology, ecology makes it really easy for me to ask you scenario questions or case study questions. Describe a set of circumstances and then ask you about that. Okay, so do not be very surprised when you see a ton of questions like that style, okay? So it lends itself really nicely to those types of questions. So you'll see some of those. All right, so this is a good example here in the middle of a random species distribution pattern. What does it mean to be random? Is there any actual pattern that's discernible? Not really, it's random, right? It means wherever. So dandelions are a good example of a random distribution pattern because they're wind dispersed species. So you see this frequently in these types of organisms. They germinate when they land in a favorable habitat. That is random, right? So if you look at this graph, your sort of picture of representing a random species distribution pattern, there's no rhyme or reason to it. There's no discernible pattern. They're just wherever. If they land on a driveway, they don't germinate. They land in a field, maybe a lot of them germinate, right? And how do these get around again? How do these seeds get around? Wind, they're wind dispersed. You guys know what happens to a dandelion when it matures and it makes seeds? Does it look yellow anymore? What does it look like? White fluffy stuff, right? What's the fluffy stuff for? Carry them on the wind, right? So that's literally where as the wind blows, right? It doesn't get any more random than that. So that's a random distribution pattern. There's also nothing there's no like intraspecies competition between these individuals necessarily. A lot of them can live in the same place. So you don't have any sort of extenuating environmental circumstances that's causing these things to live in any other way. So that's different than when you see a uniform or a clumps distribution pattern. So uniform means there is a pattern, and look at that pattern. What do you notice about that? It's even, right? Evenly spaced. So what can you make? What kind of a conjecture or prediction can you make about a species that displays a uniform distribution pattern? Territories. Yeah, absolutely. So you can see this happen when plants secrete substances that prevent other plants from growing near them. Okay, so some plants will do that. They actually put chemicals out into the soil that make them have like a boundary around themselves. They exclude others, so they decrease competition. Or territorial animals, those who defend a particular area, right? Penguins are a good example because these dudes are super territorial when they're breeding, okay? But when they're not breeding, they're pretty social. Okay, so your species distribution pattern is not necessarily fixed, meaning it can change from season to season, right? From month to month, depends on what's going on with that particular species. Because remember, populations are dynamic, yes? So that doesn't have to be the way that it always is, just because sometimes they act that way, right? Because species are going to change their behavior, but that's what uniform means. And then what do you notice about clumped? What does clumped mean? It means clumped, right? That's why the terminology in these chapters is so nice, because it means what it means. It means that they congregate, they form groups, schools of fish, herds of elephants, right? What else can we think of that might be clump distribution? What about people? Have you ever looked at a map of urbanization? Most people tend towards cities, right? High population density in cities. And as you get out here into the suburbs and beyond, into rural areas, what happens to population density? It decreases, yeah. So we definitely exhibit clumped distribution patterns if you look at human populations too. So this is going to happen mostly when you have social species. Okay? If you fight with each other, that's not going to work so well as a strategy. All right, so this is pretty straightforward, right? Nothing too confusing or horribly difficult. Okay. Let's look at range maps as promised. So remember what range is. What's range? Yeah, where you find the species. So what do you think a range map is? The map that shows where they're found. Yeah, see? Okay, perfect. So range maps are going to just indicate visually where in the world you find something. So something like a penguin range map here shown in red shows you that all the penguins that are naturally occurring are going to happen in the southern hemisphere. And polar bears exhibit a northern hemisphere distribution. That's all it is, you guys, just range maps. The interesting thing, though, that is important that we haven't talked about yet with range is that you have to include anywhere that you'll find an organism at any point in its life. So if you're talking about things that migrate, right, like certain species of birds or insects or anything that moves from one place to another, you'll include all of its entire range that it's anywhere it's been any time. So if you're looking at like a bird range map, an occurrence range map, notice that you have non-breeding down here, you have migration here in yellow, and then you have breeding season here in orange. Okay, so this entire thing is considered the range of the ruby-throated hummingbird. Even though in the summertime you'll find them up here, in the wintertime you'll find them down here, and as they're moving in between, you may see them skirt over into the Midwest. Right? So all of that is included. Make sense? Pretty straightforward, right? Kind of very quiet today. Even more so than usual. All right, let's talk about demographics. Interestingly, before we really get into it in too much detail, let's look at this last bullet. This is a life table. Okay, so we'll talk about what this means here in just a second. Excuse me, as far as demographics goes. But this is the part I want to start with. Modeled after tables used in insurance industry for human life expectancy. So demographics, what does demo mean? You guys know? Like democracy? People. Yep. And then graphics is just measuring stuff, right? So this is how we measure people. Okay, but we can use the same methods to measure populations of organisms that are not Homo sapiens. Okay, so this field of ecology, demographics, is modeled after the way that we look at human populations. And why does the insurance industry care about things like human life expectancy? What kind of insurance might care how long you are expected to live? Life insurance. Why? Because they pay you when? When you die. So they're calculating how many years, how many months are you going to pay them before they have to pay your family, right? So that's what life tables were originally designed for. And if you look at what you're seeing here, this one is for a Dall Mountain sheep. Okay, so nobody's doing life insurance for Dall Mountain sheep. So this is clearly an ecological application. But it's definitely built around the same concepts that you would look at with human beings. Things like birth rate, death rate, life expectancy. So this first column here is just age interval. okay, in years. So you've got 0 to 6 months, 6 months to a year, and so on. Okay, so that's pretty straightforward. Column B, number dying in age interval out of 1,000 born. So out of 1,000 Dall Mountain sheep that are born, 54 out of 1,000 will die between the ages of 0 and 6 months. And that's what this is telling us. Then you have numbers surviving at beginning of age intervals. You can look at the same question in different ways, mortality rate, and then you get mean life expectancy at each age interval. And so you can sort of see the applicability for life insurance companies, right? So if you have to insure a Dall Mountain sheep that is 3 months old, the chances that that individual will die in that age interval, 54 out of 1,000. Does that make sense? What about between 6 months and a year? What happens there? Now that number goes up by almost 3 times, right? So you're 3 times more likely to die in the age interval from 6 months to a year than you are from 0 to 6 months. That's interesting, right? So if you are an insurance agent for sheep and you're trying to put a policy together for a 3-month-old, is it going to cost less or more than a policy for an 8-month-old? It costs less because you're less likely to die, right? And they're less likely to have to pay out. So you guys see the tie-in, right? But then look here. If you make it to a year old and you're a sheep, Dall Mountain sheep, what happens to that death rate or that number of individuals dying in that interval? Drops by a lot. And then it holds pretty steady between 2 and 3 and 3 and 4. Then around 4 years, it starts going up a little bit. By 7 or 8 years, it's higher. Look at this. Between 8 and 11, it jumps back up again, pretty high, right? So what can you say about the life expectancy of a Dall Mountain sheep by just looking at these numbers? How long do you think the average sheep lives? Say again? Yeah, between 8 to 11 years probably, right? Most of them are dying at this point, unless they died very young. So what can you say about infant mortality or juvenile mortality? It's higher. Why? What do you think? Why is it more likely that you'll die from 0 to 6 months? You're young and you're helpless and you get to feed yourself and you probably can't get your own food, right? You're still dependent upon your mother, likely, the sheep, right? Then what do you think happens here from 6 months to a year? I don't know that much about the biology of Dall Mountain sheep, so there's no real right answer. We're just spitballing. Yeah.
Maybe they start transferring from their mother's care to their own individual care, and so their life expectancy goes down.
Yeah, they start to branch out, right? Maybe they're getting bred. Maybe they're starting to wean and move away from mama and look out for their own food, but they're not really like totally mature yet. So maybe they're less savvy at avoiding predators, right? Maybe they're not that good at finding their own food. Maybe they fight with each other for territory. I don't know. But you can sort of see the types of questions that you can then ask about a population once you know these types of information. Does that make sense? So it's not just like we're so interested in how to sell life insurance to sheep, but that's clearly not the application here. But you can look at these and start to ask those types of questions. And then you can ask questions like, is it food availability? Is it competition? Is it predation? And then you're looking even more in depth at the ecology of these populations. Does that make sense? Cool. So that's what I really want you to understand about demographics. And we'll talk about some of these values going forward when we look at life history strategies and stuff like that. How are we doing on time? We're doing pretty good. Okay. Let's look at survivorship curves. Told you the graphs, so we'll keep on coming, right? So these are pretty straightforward though, okay? Not too complicated graphs that you have to read. So survivorship curves are showing just number of individuals surviving at each age over time. So you're basically taking this information that we just looked at for the sheep and putting it on a graph, okay, and tracking it with a curve. So you've got type 1, type 2, and type 3. So I'm gonna ask you guys to tell me which is which. So you're gonna have to be able to look at a graph or read an example of an organism and how it acts and tell me which one it fits. So type 1 is mortality is low in early and middle years and occurs mostly in older individuals. Here's type 1. This is time on the X, number of individuals surviving on the Y. Again, it's a log scale, so you get nice curves and straight lines you can sort of look at and interpret. So if you have a high number of survivors early, and it's pretty steady, and then towards the end, you see a steep drop off, what can you say about that type of survivorship curve? What can you say about infant mortality? Low, because survivorship is high, or right? What can you say about juvenile mortality? Pretty low, right? You still have high survivorship. What about adults? You're still doing pretty well. And then what is probably a number high on the list of number 10 causes of death? Old age, yeah. So here's us, right? We would fit a survivorship curve for type 1. Now, is this every single individual is going to follow this same pattern? No, there are going to be exceptions, obviously. This is just an average, right? But we have really high juvenile and infant rates of survival now, right? Especially with modern medicine, right? Infant and juvenile mortality has come down a lot since we found things like antibiotics and vaccines for childhood illnesses. So that's type 1, okay? We're going to talk about some other factors that lead into these survivorship curves as well, but we'll get to that in just a few minutes. Type 2 is here, the red line. Type 2 example organism that's given here is birds. Mortality rates are fairly constant throughout the lifespan. Death is equally likely at any age, okay? So if you're a bird, the chances that you're gonna die are pretty much the same, whether you never hatch, right? The egg doesn't hatch, or something eats the egg, right? So you could be predated out of the nest. You could hatch, but then fail to thrive, fail to compete. There is a lot of siblicide in bird clutches, so where like one will actually push the other one out of the nest. It's pretty brutal stuff. Maybe you can't fledge, like maybe you try to fly and you fail, right? And something eats you because you get too close to the ground, or you just fall and hurt yourself and die. Or you make it to be an adult bird and then a hawk gets you, right? Or you fly into the side of a building, yes? Or you fight for territory with another of your own species and fight to the death, right? So there's lots of different ways for it to be dangerous to be a bird, but it's equally likely throughout your lifespan. So there's no real pattern like you see in a type 1. You guys see the difference? Makes sense? All right, so what do you think type 3 is? It's the green one. It's the exact opposite of a type 1, okay? So survivorship is high in the very beginning, and then what happens? Drops off really quickly, okay? So what does this mean? Mortality is highest in early life. So really high infant or juvenile mortality. But if you survive that phase, you are likely to live a relatively long period of time. A good example is given of a tree. So when you are a tree and you are reproductive, you are making seeds, right, to try to make new trees. How many seeds would you say an average tree can produce per season? Like 2 or like 100 or like 1,000? Probably thousands. Now, there's going to be some exceptions. We'll talk about that. Like palm trees put a lot into one big coconut, right? But something like an oak tree or a pine tree, I mean, seeds everywhere, right? So how many of those seeds do you think actually turn into full-on big adult trees? 3? Yeah, like 3. Exactly. Very low, right? Low percentage of the overall production. So you have really high number of offspring with the full understanding as a tree, as much an understanding as a tree has, right? That they're not all going to make it, right? So you invest a little bit in a whole lot of offspring and hope somebody makes it. But that somebody that makes it and germinates and grows up past the sapling stage and doesn't get mowed off, eaten eaten or mowed or whatever, becomes a big tree, then you got a pretty good chance of making it, I don't know, 100 years, right? If you make it to an adult tree. So it's kind of the opposite of well, it's the exact opposite of a type 1 curve. Marine invertebrates is another good example that your textbook gives, because marine invertebrates tend to produce tons and tons of offspring. So like a jelly, for example, is going to make thousands of babies, thousands of larvae, but those larvae are just swimming around in the ocean. So what do you think happens to most of them? They get picked off, they eat them, right? They're food, they're plankton, essentially, right? But if you don't get eaten, you grow to a full-on jellyfish, then maybe your life is great, right? For the lifespan of jellyfish is probably like a year, I don't know, for certain species. Anyway, so what we're going to look at here too, the next topic we'll talk about is fecundity. So we'll get here in just a second, but fecundity has to do with how many offspring you can make at a time and through your lifespan, okay? So something like a tree or a jelly that's making thousands of offspring, but not expecting the vast majority of them to survive, has a totally different life history strategy than something with a type 1 curve. So think again about primates like us, right? How many offspring do you generally have at a time, a human? Like 1, mostly. Sometimes 2, sometimes you have multiples, but average 1 at a time. And then how long do you take care of that 1 offspring? Sometimes like 30 years, right? Maybe 18, right? And then you're considered an adult, and then you can go off on your own. So our parents spend a really long time taking care of us. When you're first born, look at this, your survival rate, you got a pretty good shot at making it, right? Why? Because somebody is like this all the time, right? You can't walk, you can't talk, you can't get your own food. You're basically your own worst survival risk. But there's parents that are taking care of you and making sure you survive, right? We invest a ton of energy and resources and time in our offspring. That's our life strategy, right? We make 1 or 2 or 3. What's the average number of kids in an American family? You guys know? 2.5. So you have like half a kid and then you're done. No, 2 or 3, right? Is generally average for an American family. So you stop at a certain point when you think, okay, I don't have enough resources to go around and I need to stop now. This is my family, right? Because you're investing so much energy, time, and resources into making sure that those individuals survive and thrive. That is a strategy. You don't have to make 1,000 offspring, because the ones you do have are going to have a really good chance of making it because you are investing a time into those individuals. Does that make sense? Also, I'm going to go ahead and forward to the fecundity slide, so we'll start talking about it in terms of this. We're looking at life history strategies here. A life history is not just reproduction, but that's sort of what we're talking about here, but it also includes all of the events over the lifespan of an organism, like how you use your resources. Remember on Monday when we talked about resource allocation? So first you do resource acquisition, which means what? You get them, you collect your resources, right? And then allocation is how you spend them once you have them. Okay, so we're looking at that in terms of reproduction here. But you're also doing things like growing, right? You have to use energy to grow your own body tissues and maintain, repair, replenish, things like that. So your energy budget is how you balance your energy intake with the use of your energy. Do you use it for metabolism, to reproduce, to take care of your offspring? Do you store some? Okay, so this is basically how you allocate your resources. That's your energy budget. It's just like your money. Same thing, right? How you use what you've collected. And then, as I mentioned before, fecundity is the potential reproductive capacity of an individual. So how many offspring can you make as an individual through your entire lifespan? And maximum fecundity, when we measure fecundity, we're talking about how many offspring could you produce ideally, meaning perfect circumstances, if you had as many offspring as possible every time you reproduce, and you repeat your reproductive cycle as soon as possible after you have offspring. So a human being's maximum fecundity is probably like 20 or more, right? But we don't do that, okay, because that's part of our life history strategy. But that's what fecundity means, is how fertile or how reproductive, what is your reproductive capacity? That's what fecundity is. In animals, like what we're talking about with humans, fecundity is inversely related. What's an inverse relationship again? One shows up, the other goes. Yeah. So inversely related to the amount of parental care given to an individual offspring. What does that mean? Maximum reproductive capacity goes down when parental care goes up, because you're spending your energy budget on one offspring at a time, or 2 or 3 max. So we're not even necessarily talking about parental care after that offspring is born. We're also talking about investment into the quality of the offspring, okay? So you have an energy budget, and how you choose to spend it depends on the organism, and depends on the best life strategy that you are adapted for, right? So if you are a pine tree and you're making 1,000 cones, and in each cone you've got 50 seeds, each one of those seeds, you put some energy into making it, right? It costs you some of your metabolic energy and some of your resources that you've stored up to make that baby, to make that embryonic pine tree, give it some endosperm in there to eat, like healthy nutrients inside the seed coat. But you made 50,000 of them, right? So each one got a little bit of your energy budget. But if you are a human and you make one infant and it takes 9 months, 40 weeks to grow it, how much energy is put into that one offspring? A lot, all of it, right? So have you ever heard the term all of your eggs in one basket? You're taking all of your reproductive capacity and you're funneling it into this one offspring. So it makes sense that you would take care of it throughout the first 5, 10, 15, 20 years of its life, because that is a huge investment of your resources and your time and your energy. Does that make sense? Now, if you're a pine tree and you make 50,000 seeds, you are investing 0 parental care after the seed disperses, right? Does the pine tree look after its pinelings? No, right? Of course not. So it's a completely different life history strategy, but fecundity in plants is inversely related to number and quality of seeds. So the same concept exists. If you are a pine tree or an orchid, so the orchid example is a good one because orchids make like thousands and thousands of dust-like seeds in a single capsule. Okay, so flower, one of these orchid flowers will eventually make a seed capsule like this, and that looks like dust. You can see that, like every single one of these little particles in this picture is another orchid seed. That might be 1 million. You counted them all, right? So you've got a ton of seeds. That is going to be high fecundity. You guys with me? High fecundity, millions of seeds. Okay, but each individual seed is kind of expendable if you look at it as an individual. What are the chances that any one of these is going to make it? 1 in 1 million, right? But if you look at something like a palm tree, they're making coconuts. A coconut is a huge investment of resources to make a single baby palm tree. That baby palm tree lives in here. All of the water and milk, all of this white fleshy stuff, that's all food for that one embryonic palm tree in there. And then you have a tough outer seed coat, and then you have a husk, and then you have another seed coat that helps this thing float to its ultimate destination. And how many coconuts do you see at a time on a palm tree? Not that many, like 10, maybe? Not 1 million. You guys see the difference? So coconut palm has low fecundity because high quality of seed. A lot of resources went into making that coconut, which has 1 baby in it. How much of this orchid's resources went into a single orchid seed? Not a lot, right? A very small amount because it made so many. Do you see the difference? Then you can also tie that back to survivorship curves. You can explain the survivorship curve based on fecundity. Parental investment, parental care, offspring survival, it's all sort of interrelated. Make sense? You ever think about that before? Ever think about how much energy and time and resources your parents put into making you human? Thank your mama. Huh? All right, one more thing about reproductive rates, and we'll stop for today. We only have about 5 minutes. Oh, let's see. Now let's just stop. How about that? Because there's 2 more slides. 2 more slides on population models. So yeah, we'll just stop with fecundity. I think that's enough for today, even after